Okay. I'm Rae. I'm an ACVNU resident at Friendship and will be presenting Journal Club today. This paper's a little bit shorter, so it probably won't go the whole time unless people talk about things. So feel free to stop me. So the paper I'll be talking about today is Apoptosis Inhibitor of Macrophages in cats, a potential link between an exon 3 variant allele and progression of naturally occurring chronic kidney disease. And this article is from JVIM in May of 2025, so just recently published here. And so in the introduction, they talk about the clinical course and prognosis in cats with CKD and how it's variable, that after an insult, just like we all know, they could have progressive loss of GFR or stable GFR show improvement, and how genetic influences can influence that response. And so then they bring up AIM, or apoptosis inhibitor of macrophages, and how this is a protein that's important for kidney repair. And they know that from human and mice studies. And there have been studies that show that mice that are AIM deficient have then progressive renal dysfunction. And so, a little bit more from the introduction on genetics, because I definitely did not remember all of these genetic things from undergrad. They show this figure and they go through that there's a gene that's organized into exons and introns, and DNA is transcribed into messenger RNA, so the splicing out of the introns, and then that messenger RNA will contain a sequence of exons that codes for specific amino acids and then proteins. And in cats, what we know is that there's a wild type feline AIM protein that has 3 scavenger receptor cysteine rich protein domains. And then there's also a variant feline AIM protein that has 4 of the SRCR domains. And so they show it here that exon 3, exon 4, and exon 5 correlate to those 3 domains. And they hypothesize that this change in the variant AIM was due to a duplication of exon 3 in the cats. And so that would then create that 4-domain protein. And then thinking about if that variant AIM allele would lead to risk of further decline of renal function. And so overall, their hypothesis was that cats that had the variant for exon 3 allele leading to that variant AIM protein, so the 4-domain, would experience impaired kidney lesion repair, and that would be reflective as those cats having a more progressive decline in their kidney function. And so overall objectives were to confirm that that domain duplication was from a duplicate of exon 3, and then to see if that was true, if those cats with the variant type were at higher risk for decline as compared to the cats that had wild type. And so just to lay it out really clearly, we have the wild type 3-domain AIM protein that is associated with normal kidney repair, and then the variant 4-domain AIM protein that is suspected could that lead to impaired kidney repair. And so that's kind of the basis of the introduction for this paper, and it just left me wanting to know a little bit more about AIM before I read the rest of the paper. So we're just going to take a minute to kind of touch on this paper from 2016, Impact of Feline AIM on the Susceptibility of Cats to Renal Disease, just because I needed more information to kind of understand why do we care about this. So again, AIM, apoptosis inhibitor of macrophages, it's a circulating protein made by tissue macrophages, and during acute kidney injury in humans and mice, the AIM will dissociate from IgM and then appear in the urine, and so it'll be filtered and that will interact with KIM1, which then promotes engulfment of debris in the dead epithelial cells in the proximal tubule, and that promotes renal recovery. And so they looked at studies in mice and found that mice that were AIM deficient, that were then subjected to an ischemic acute kidney injury, had decreased debris clearance and persistent renal inflammation, and then higher mortality as compared to wild type mice. And then mice treated with recombinant AIM after an ischemic induced acute kidney injury had removal of that debris and amelioration of their of that pathology that they saw. So um, they also looked at mice that had the normal wild type AIM, and then felinized mice, so mice that express the feline AIM, specifically the three domain, I believe they looked at with AKI, and found that these felinized mice had higher creatinine levels, and none of those mice survived. And then they also looked at histology and found that on day 3 post AKI with wild type AIM, they had less intraluminal debris and more recovery of the brush border as compared to those felinized mice. And then the acute tubular necrosis scoring was also higher in the felinized mice. So that's just what's shown in these figures where you can see it clearly. The wild type, their creatinine kind of plateaus and then see some improvement. The felinized mice have progressive elevations in their creatinine. And then for survival, none of the felinized mice survived. And the wild type had greater survival. And then with the acute tubular necrosis scores as well, are higher in those felinized mice. And so then they talk about AIM in cats. They found that there's higher levels in the serum as compared to humans and mice. And they also found that there were 2 types. And so the 3 domain and the 4 domain correlated with those different sizes. So the 37 kilodalton is the 3 domain, the wild type, and the 49 kilodalton is the 4 domain or the variant. They also talk about how AIM doesn't dissociate from IgM in cats during AKI like what is seen in mice and humans, and could that contribute to progressive renal decline? When there is an AKI, AIM is supposed to dissociate from IgM and then accumulate in the urine interact with KIM1 with the intraluminal debris and promote clearance. And so when they looked at cats with AKI, there wasn't IgM free from AIM, and they suspect that there's higher binding affinity for feline AIM with IgM. And so this is just a schematic that I stole from online. Dr. Miyazaki, who was on the 2016 paper, and I think looking at giving recombinant AIM in cats in Japan. I just thought this was a nice visual on why maybe we should care about AIM, and showing that in human and mice it dissociates here, and then in cats that there's less dissociation. So basically, then the question being, would giving recombinant AIM to cats that have acute kidney injury, would that be beneficial? And that's kind of what they looked at in felinized mice. So if they give a felinized mice recombinant AIM, then that ameliorated the AKI, and could this be a potential therapy? So again, with these figures, you can see that there was higher survival of the mice given recombinant AIM, and their creatinine plateaued and then showed improvement, and that their acute tubular necrosis scoring was lower than those that weren't given the recombinant AIM. So a little bit of a detour from what we'll talk about with this paper, but interesting to know a little bit more of the background. So again, specifically in this 2025 paper, the question being, if cats have the wild type versus the variant allele, does that alter the function of the AIM, and do we see that as progression in their kidney function? So for their study design, they Evaluated medical records of cats with CKD from 2011 to 2022, and they had exclusion criteria. So, cats that were diagnosed with other renal conditions, cats with abnormal T4, or that lacked follow up data, or cats that were dehydrated or on fluid therapy or receiving drugs that could alter the serum creatinine concentrations were excluded from the study. And then for inclusion, they did have to have CKD. So that was defined in this study as a creatinine greater than 1.6 for greater than 90 days with a USG of less than 1035, or creatinine greater than 1.6 on 2 occasions within the 90 days. And then they needed to have greater than or equal to 180 days of follow up for their creatinine. And so for genotyping, they looked at the number of copies of exon 3. And so if they had 2 copies, then that corresponded to the wild type. If they had 3, that corresponded to being heterozygous. And then 4 was homozygous variant. And for their phenotype classification, they used 2 different approaches. So, approach A was based on iris staging. Cats were put into their iris stage, and then over the follow up period, if they advanced a stage, they were defined as progressive, and if they didn't advance a stage, then they were defined as non progressive. And then for approach B, they just looked at serum creatinine. So, if they had a less than 20% change, they were non-progressive, and if they had greater than 20% increase in their creatinine, they were defined as progressive. And so for the DNA isolation and sequencing, they talk a lot about the technical aspect of this that I won't get too far into, but essentially they sequenced the DNA and amplified the AIM protein and visualized it on a gel. So based off of the expected molecular weight, was it the 37 kilodalton 3-domain wild type, or was it the 45 kilodalton 4-domain variant? And then they did nucleotide sequencing of both and converted those into amino acid sequences. And so based off of that, predicted what the tertiary structures for the AIM proteins would be. And so you can see that the left is the 3-domain wild type and the right is the variant protein, so 4-domain due to the duplication of exon 3. And then they discuss how, suspected potentially the variant could have altered function because it's larger, it's more negatively charged and less water soluble. So could that lead to, this structural difference lead to a functional difference that alters the normal kidney repair reflected as progressive kidney disease? And so this is just a figure from the supplemental material. If you're more visual, A is showing the homozygous wild type, so just one exon 3. B is showing the homozygous variant, so there's 2 exon 3s. And then C, you can see both. There's the 1 exon 3 and then the 2 exon 3s. And so for their cases, they had 172 medical records of cats with CKD that were found, with 91 having archived DNA, and 50 of those cats ended up meeting the inclusion criteria to be included in the study. They had overall 24 cats, so almost half, that had progressive worsening of their creatinine, and 29 out of 50 that stayed within their same iris staging. So for the staging progression, they had 24% or 12 cats that progressed from stage 2 to 3. They had 2 cats that went from stage 2 to stage 4. And then they had 4 cats that improved from stage 2 to stage 1, and 2 cats that ended up improving from stage 3 to stage 2. And so overall for the AIM protein, 30% of cats were homozygous wild type, 52% of cats were heterozygous, and then 18% of the cats were homozygous variant. And so for the results, there was a significant association between the AIM exon 3 copies and worsening of creatinine. And so I really liked this figure, how it shows the dashed line is the 20% cutoff, and you can see a few of the cats were progressive with the homozygous wild type. A large proportion of the homozygous variant were progressive, and then about 50% of the heterozygous had progression. And so, it appears that just comparing the homozygous variant to the homozygous wild type, there is an association with worsening creatinine with the homozygous variant. And here is just another figure from the paper that's showing it based on their approach A, so based on staging. And again, you can see that the wild type cats, there's one that progressed from stage 2 to stage 3. And then for the homozygous variant cats, although there's less of them, only 3 were non-progressive based on their stage. And then, some of the heterozygous variant cats had progression. And so, kind of to tie their results together, table 2 shows that with the homozygous variant, specifically compared to the homozygous wild type, there was worsening of the iris stage as well as worsening of the creatinine. And you can see that the odds ratio is 1.55 to 196 for iris stage, or 1.38 to 67 for progression of creatinine. And so greater than 1 indicates that a homozygous variant is more likely to have progressive kidney disease as compared to the homozygous wild type. When they looked at the homozygous variant with the heterozygous variant and compared that group together to the homozygous wild type, then you can see that the odds ratio crosses over 1. So not necessarily statistically significant or more likely just because you have, or the cat has one allele of the variant doesn't necessarily mean they're more likely to have progressive renal dysfunction, is how I interpreted this table. If anyone has anything to add, let me know. Okay. And then for additional results, they also looked at the frequency of the AIM exon 3 genotypes in 100 banked feline samples and had 38% homozygous for wild type, 42% heterozygous, and 20% homozygous variant. And so in total, 62% having 1 or 2 copies of the variant AIM allele. So there is less of the homozygous variant type, and I wondered, could that mean there is a degree of selective pressure against that variant allele, or is this just genetic drift? Because it is a small sample size, there's only looking at 100 feline samples. And so I went back to genetics and looked up the Hardy-Weinberg equilibrium. Based on that equation, it told me that maybe there is higher than expected frequency of the homozygous wild type and lower than expected frequency of the heterozygous genotype, about what you'd expect for the homozygous variant based off the allele frequencies. And so maybe could that indicate there's selective pressure against the heterozygous? But again, this is such a small population. I don't think we can say we're necessarily in equilibrium and that this is truly due to selection pressure. And based on what they found in the study, there wasn't a statistically significant difference with the heterozygous. So maybe that is just too small of a population to look at, and I'd be curious to see it in a larger population of cats.
Hey, Rae.
Yes.
In your deep genetics research that you've done for this, which far exceeds my knowledge, is there any reason that it makes sense that the heterozygotes and the homozygote variant ones have gained the extra segment how do we know that for sure versus what if the what was we're being called the wild type, what if they actually have a deletion in a segment, right? Like, how do we know what normal is if the minority of patients that are seen in this population have wild type? Does that make sense?
Yes. I guess I don't know the answer to that.
I thought most of it was because that's cross species, it doesn't have duplication. So if you compared it to like mouse and rat humans. Does that make sense, Judy?
Okay, so the AIM protein across species, majority of species are going to have just the three segments in the protein. And then if they have the duplicates, they're the outliers. Is that right?
That was my understanding from this entire concept. However, I've not read it in depth.
Gotcha. Thanks.
I wish I had the answer for you, but okay. Overall, cats with homozygous variant type were more likely to have progressive decline in kidney function compared to homozygous wild type. And again, they speculate on why this might be the case, that the four domain protein is larger, has a higher negative charge, could hinder renal filtration. I do think that in the 2016 paper, they had touched on the three domain and four domain, and that both were filtered into the urine. So potentially, does it still reach the tubules? Does it have decreased interaction with KIM1? Or maybe we don't exactly know. Maybe we need a larger study. Um, they do bring up that of the 24 cats with progression in their CKD, 20 of them, so 83%, had at least one of the additional exon 3. So either were heterozygous or homozygous, but again, it's a rather small population. And so I think this just opens it up to what is next. Is there impairment of filtration based off of the genotype? Could this be utilized for prognosis? Could we give cats AIM and improve outcomes? And I think maybe that is being looked at in Japan. I don't know if anyone knows. I was like trying to do a deep dive on if they are actually enrolling cats for recombinant AIM, but I couldn't find a definitive yes or no answer. I don't know, have others been asked about this by owners before or have anything to share? No. Okay. Or what if, is there a way to improve dissociation of AIM from IgM in cats? Would that be just as helpful as giving them AIM? So I'm interested to see what happens in the future. That is all. I know that was really short. Does anyone have anything to add?
Ray, have you come across anything that looked at the dissociation between the different AIMs and IgM? Because you said that in people and in mice, it does dissociate, but in cats, it doesn't tend to. Is if they have one of the mutants, is it more or less dissociable from IgM?
I don't think it was based on the 2016 paper. They didn't say that I remember that there was a difference between them. Um, but I think that warrants further research. And it wasn't like the main point of this paper, at least. It didn't really come up.
I'm wondering if anybody else has had clients that have asked about this or have started trying to purchase some of the supplements. I think the main researcher from Japan is associated with a company that now makes several different types of supplements for cats with CKD. And I don't know if anyone has actually seen any of these cats coming in with on these products or if you had clients that have asked about it. I've had a couple clients ask personally, but it's not been something that's come up very commonly amongst the people who are coming to see us.
Yeah, I haven't had anyone ask me about it, but then when I was doing more research, I did find like a capsule um, from the researchers on the 2016 paper, and it didn't like say what was in it, but I don't know. I was like, maybe this is the cure. I need to buy it.
Ray, a couple people responded to the
Oh.
question in the chat.
Oh, great. I'm not sure how to see the chat from here. Should I can stop sharing.
Oh, no, it's all right. Basically, they said yes, that their clients are asking about it.
Oh, okay.
So it looks like Texas and North Carolina.
What are you guys telling them then?
I know nothing. And we, as you mentioned, the I saw something, I think probably the same thing as far as some capsule. I don't know what it is, what it does. I don't have any data support giving it. So I have discouraged the use personally, rightfully or wrongfully, of how I classify weirdo supplements, if I don't know what's in them. And I have a client right now trying to get her cat to Japan. So
Do you know then? Are they actually enrolling right now?
I didn't look into it. I'm sure I could find. I'm sure I could follow up with you. It's a very eccentric cat owner of mine who has added me to her will to take care of the cat, and so I'm sure I'll hear a lot more about it. But she was actively trying to get her cat to Japan for this clinical trial. But
Ray, that sounds right up your alley.
The one
Ray
client
is
that I had,
also a very eccentric cat owner, for anybody who did not know.
I had one cat owner that I was trying to get into the trial in Japan. Last I heard from that owner, they were not currently accepting patients for it. I haven't had anybody else like really try that hard to get it. And since we have a bunch of other studies going on, they've kind of gone more that route.
What I don't understand about the physiology of adding humanized or non-felinized AIM to a cat's body is that if the problem is that the AIM is too sticky and plugs up, binds to debris, and does not disassociate, you're not getting rid of the feline AIM, right? Like you're just adding more protein to the mix. So are you really ever going to break up that dissociation that's already too tightly bound that gets getting stuck in the tubules? And I don't feel like, as much as I read about this, which is only briefly for writing textbook chapters and stuff, it just doesn't answer that. Like we're not getting rid of the actual problem protein that's like clumping up stuff in the tubules. Does anybody else have any thoughts about that?
No, Rachel, I had the same consideration for that. And the other thing that I always get a little bit freaked out with foreign proteins too.
Yeah.
And so I don't know, well, and I guess we'll see if this group ever publishes it. But I would be really terrified of adding some foreign protein to the mix, and these cats with failing kidneys who already have mucked up tubules
and already have a lot of inflammation in there anyway. They've already,
Yeah.
the inflammatory cells are there and already primed and turned on.
Yeah,
And you're
I'm
adding, yeah.
waiting for some mega GN or some other horrible, hypersensitivity reaction. So yeah, I agree. I had the same thought. I said, well, I could understand if you were giving like gene therapy and you were actually altering those three, make them back into a 3-domain cat or something like that. And that would be different. But just giving them an extraneous protein, I don't understand how that makes all of this go away.
And having a genetic marker would be nice to be like, do we have a genetic tag to say that this cat has a higher risk of progressing than a cat that was homozygous without the duplication. So maybe they're gonna plateau and probably be fine. And so I think having having a genetic predictive test would be helpful, but I just don't understand it as a therapeutic. So
I guess I thought that the IgM and the AIM protein were bound in the serum, and then it dissociates so that the AIM can filter into the tubule and bind, so that if you're just giving AIM, it's filtered into the urine. I thought that was my understanding. I'm not, maybe that's incorrect.
I guess I'm just, no, I think that is right, but it's how does that extra protein knock off? Like, does it knock off the one that's already, like the felinized one's still being made
Yeah.
by the cat.
But does it, since it's not feline, does it bind to the IgM and then it dissociates and goes into the tubules, and the feline one is just not doing that, like not doing
I
what
think we
want?
don't know that.
Yeah, that's what I think that's not clear, is like, is it going to be more attracted to the extraneous non-felinized one? Like, we don't know any binding assays. We don't know any of that, like stoichiometry, that's the actual basic chemistry of how this would work, hasn't been demonstrated yet. Or at least not published. It very well might be known, but it's not published, at least not to my knowledge. So I find it to be a fascinating topic, and I think it is very interesting to say that this would be a risk factor, but I'm not entirely sure at this stage that it has I can't see how you're going to make it a therapeutic. But
And I think from this paper, this paper today that we're talking about, the main one, actually could be helpful for designing clinical trials in cats with CKD. Because when we look at the risk factors that we have for CKD, most of them are associations, and it makes sense that cats with worse disease have the risk factors, that they're going to be more hyperphosphatemic, have a higher FGF23, that they're going to be more anemic, that they're going to be hypertensive. It's that all could be just cats that are on a later spectrum of disease. And that doesn't mean that those are necessarily the players that drive progression. Because we don't have studies that show if you correct anemia or hyperphosphatemia or FGF23 or anything, that you actually take away the increased risk for mortality that those signals had. And those studies have just not been done, but we would need them to show if they are actually therapeutic targets. And we treat as if they are, but we have no evidence for that. Where it's possible that this genetic marker having an association and presumably being there well before the onset of any kidney disease, if it was associated with, if injuries might have occurred, that these cats are more at risk of having maladaptive repair and maybe having more progressive disease. And you could argue that might even get you to what is called stable CKD and not just progressive CKD, but you had to get there to begin with. But perhaps in designing clinical trials where we're trying to look at therapies that could have an impact on progression of disease, we could look for those cats that have at least one, if not two copies of this mutation, because they would have a higher risk of having progression, which means that you could actually enroll fewer cats. It would have better power in actually predicting something.
And just think about having a litter that has heterozygotes, like having a mixed litter and having your age-matched controls that are otherwise genetically very similar. And the only difference, or one of the milder differences, is their AIM protein, and following those litters. Like, that sounds like we would have a lot more power in the data and just seeing what they do.
Yeah, exactly. I mean, right now we try, rightly or probably also wrongly, when we're enrolling patients in clinical trials, we're looking for those that lack so many of the factors that are associated with them not doing well because we want them to be able to complete the trial, and we want hopefully the only factor that is being tested or attributable is our intervention. But at the same time, we're probably selecting for those patients that have a lower risk of progression from the beginning. So we're likely to have underpowered studies where we're not going to see a therapy have an outcome, which doesn't in no way prove that the therapy is not effective, but the research will probably die with that single study because it's so hard to do any research, let alone replicate research in our field. So, to me, I think that this is really interesting. And if it could be done in a fairly quick and inexpensive way, it could be considered an interesting method of enrolling patients, that if you just get rid of the wild type cats, then you are, if this population holds true to a larger population, which is certainly a bigger question that's not yet evaluated, it might make it easier to do these studies and actually be able to identify impact of therapeutics. To me, I think that's way more exciting than thinking about giving these cats it as a therapeutic. I agree with what everybody said. It's nuts. Although I can buy, for $144, I can buy the full spectrum care set, including the Dr. Toru protocol, the intensive protocol, and the probiotic protocol for my cat for CKD, based upon the research supporting natural waste clearing system and supporting the AIM system as well too. So, yeah, not too bad.
Well, it sounds like we should have these people as vendors for Renal Week then.
We try to keep the snake oil salesman to a minimum at the conference.
When I was reading it, I was sold as a cat owner with a cat of a USG of 1030. I was like, anything.
But it's interesting because there are studies that come up and you're like, wow, this is gonna be the next big thing. Like, there was the Barapros study that came out years ago, right? And you're like, holy crap, all cats are going to get this and we're not going to have jobs anymore. And then it's just like, it doesn't go anywhere, whether that's because there were unpublished studies that showed no effect or who knows what, but it's interesting to think what might come out of this, if anything. I don't know. I'm not holding my breath. I'm surprised you didn't try
I
to
think it would be interesting if this is exciting, or given the excitement and your interest in it, like it's not hard to get your cat genotyped, right? So figure out if your cat has the variant, and then you'll feel better about yourself.
Yeah, and we were actually working with the senior author on this paper.
Mm-hmm.
So we were just collecting cheek swabs from cats with CKD, where they were trying to actually look at this data, basically replicate their data for a larger population. And so we were taking cheek swabs and sending them so they would characterize it, and then that was collected prospectively, but then retrospectively they were going back and just looking at all the records for those patients. So I think that is still I think it's still happening. But yeah, I mean, they were certainly trying to do that, at least to take it to the larger population. But yeah, if it's just a cheek swab that you can get and send out, especially if it's just for that one particular mutation, once it was automated and done easily, it would probably be something that would be fairly inexpensive for a research perspective. Maybe Zoetis can add it to as their special diagnostic test they can do for feline kidney disease.
I can't say anything.
We'll have to find someone who works there and see if we can give them some nudges.
Well, thanks, Rae. I think that was really, I had dealt with this when I was doing my pathology textbook chapter. And then that paper came out after the chapter was submitted. So I was like, I had it on my to be read list, and I'm glad you went through it. So that was very helpful.
It was JD's suggestion to do this paper, and I really enjoyed it. So
Thought it was a great paper to read, and a paper that once you start flipping through it and you look at the figures, I think a lot of people will be like, oh, I don't want to read this. This is going to be, I'm going to have to read to do the reading. I don't want to do homework to understand this. So I think you did a great job of explaining it, Rae. Thanks for putting the presentation together. It was really good.
thanks.
Well, Rae, if you don't have anything else and nobody else does either, I'll go ahead and end the meeting then.
Sounds good.
All right. Thanks, everybody. Have a good day.